Nutrition. So all of you know about nutrition, the tagline, the food supply system. So generally, just you think why we need nutrition. So this is a fundamental question. So food is needed by all living organisms mainly for growth and repair and maintain body temperature. See here in this picture, we need energy for playing, sleeping, and for all metabolic activity. We need energy, not only for animals, even for plants also. In every organisms need energy. So for that, we are going to discuss about the important areas in this chapter, nutrition. So in nutrition chapter, basically we have two types. One is autotrophs, another one is heterotrophs. Living things have been classified into two types. One is autotrophs and another one is heterotrophs. What is meaning by autotrophs? Let us see here. The organisms which can make their own food materials, that are autotrophs. Example, plants. So heterotrophs. What is meaning by heterotrophs? The organisms which cannot prepare their own food materials, that are heterotrophs. Examples, animals and yeast. So what is meaning by autotrophic nutrition? So the organisms which prepare its own food and it is not depend on any other organisms. So food is prepared from CO2, water and sunlight. It is also called as a photosynthesis process. Example, green plants and green bacteria. Heterotrophic nutrition. The organism which do not prepare its own food. Food cannot be prepared from CO2, water and sunlight. All the animals and fungi and most of the bacteria. These are all under heterotrophic nutrition. Let us see regarding this, if any we have videos regarding this one. So let us see this video, autotrophic and heterotrophic organisms. So here see plants and other photosynthetic organisms can make their own food through the process of photosynthesis. Organisms that produce their own food are called autotrophic organisms. So in this chapter we are going to see the videos regarding where we require the suitable topics. So here let us see algae. Most aquatic producers are either algae or phytoplankton. These organisms are protistas. We know very well. So in cyanobacteria, if you take cyanobacteria are actually prokaryotic bacteria which can carry out photosynthesis. Cyanobacteria is very important also. And heterotrophs. So here like animals, fungi, so must ingest or absorb nutrients from the environment. So animals ingest their food. This means that they consume their food and digest as it travels through their digestive system. So here fungi absorb their nutrients from the environment. They can release digestive enzymes which break down nutrients that they can absorb. So there are also archaebacteria and protistas which are heterotrophic and cannot make their own food. So this is the video regarding autotrophs and heterotrophs. So let us see next one. So here mode of nutrition and autotrophic. Let us see here mode of nutrition. So basically the nutrition chapter. So if we classified into two, autotrophic, just now we have seen. So heterotrophics like saprophytic, parasitic and holozoic. So examples of saprophytic, fungi and yeast, parasitic, cuscuta and plasmodium, holozoic, man, dog, amoeba. So here for this also we have one video. We are going to see the video also. So in this lesson the main important topic is photosynthesis only. So what is the required material for photosynthesis? Let us see this diagram: sunlight, water, minerals. So by this we are going to make sugars and oxygen. Why it is very essential metabolic activity The photosynthesis in the sense, the first one, so life gas oxygen released by this process, and food, the entire organisms which is essential food like carbohydrate is very important. So by these two reasons, this is the most important metabolic activity. So this is the nutrition. Let us see one video about this one. So before going to this photosynthesis, let us see about heterotrophic nutrition. So if we complete heterotrophic nutrition, then we will move to photosynthesis. So this is the session. Let us learn about heterotrophic nutrition. The mode of nutrition is parasites, and saprophytes is heterotrophic nutrition. Here see two diagrams. As we learnt earlier in the heterotrophic nutrition, the organisms depend on their organisms or plants for their nutrition. So the fungal cells which do not contain chloroplast, there are two types. So one is parasites and second one is saprophytes. Nutrition in parasites. So the organisms which are depend on the other organisms for their nourishment are called as parasites. So we know nourishment means nutrition related. The organisms like plants or animals in which parasites live for nourishments are called at host. So host means the dependent. Even there are some types of parasitic plants. The parasitic plants have some special roots called as Haustoria. We know very well about Cuscuta plant. Let us see here the diagram. So Haustoria plant, how it is looking. So in these roots penetrates into the host plants. They absorb food from the tissues of phloem, tissues from the phloem and xylem. So let us see here Cuscuta and Viscum. There are two examples of the examples. Next one, the plants that obtain nutrients from non-living organic matter are called as saprophytes. Examples of saprophytes. Here the diagram we can see. Saprophytes examples of mushroom, so fungi, so aspergillus and bacteria, cyanobacteria. So these are the examples in saprophytes. Some angiosperms which lacks chlorophyll consist of special roots called as this one. These plants absorb nourishment from the humus soil with the help of these roots. So nutrition in saprophytes. So example here we can see Monotropa. And one more thing, this is about the heterotrophs. So after heterotrophs we are going to see the photosynthesis. So raw materials of photosynthesis, water and carbon dioxide. So here let us see photosynthesis. We know very well about the photosynthesis process. So it is very important metabolic activity in autotrophs. It is a process by which plants containing the green pigments chlorophyll build up the complex organic molecules from relatively simple inorganic ones. So the important reaction occurs in the photosynthesis from inorganic compounds to complex organic compounds. So here we can see it is a process. So in presence of sunlight, carbon dioxide and water can react to give a sugars along with the oxygen. See in the diagram. So this is the photosynthesis. So in the photosynthesis process, there are many scientists are discovered in this area, even though one of the equation is very well known equation, that is Wall-Néel equation. So see here, so 6CO2 plus 12H2O in presence of H nu or light. So there are some enzymes also occur in plant enzymes. So here the main important compound is C6H12O6. It is nothing but glucose or carbohydrate. So along with this compound, we are going to in this process the byproducts oxygen and water. These both are very important. So this reaction also very important. So this reaction is called Wall-Néel equation. So Wall-Néel also experimented instead of H2O, he used H2S, so hydrogen sulfide. So in that case also he also emphasized the importance of water. And next one we are going to see photosynthesis in the video diagram. Let us see here. So the photosynthesis, see here, once we look into the this video, photosynthesis. So H2O, CO2 in presence of sunlight. These are two raw materials. So it occurs in the plant. So it is like main compound chlorophyll. So it gives sugar along with oxygen. Once see the reaction. So 6 CO2 plus 6 H2O gives rise C6H12 O6 plus 6 O2. So the plant cell, here we can observe in plant cell. So chloroplast is the important organelle in the photosynthesis. So basically chloroplast have three main important parts. One is grana and another one is stroma. Grana nothing but it is a thylakoids. So here we are going to see two reactions. One is a light reaction, so another one is light dark reaction, and it is also called as light non-dependent reaction. Here dependent reaction. So the first reaction, light reaction, so we are going to see photochemical excited stage and as well as so water molecule split into the hydrogen plus OH minus ions. So here we are going to make assimilatory powers like ATP and NADPH. So by this we are going to make, or we are going to prepare carbohydrates like sugar with the help of CO2 in the process of dark reaction. This is a main mechanism involved in the photosynthesis process. So this is the mechanism of photosynthesis process. One more video we have about the light reaction and the dark reaction. Let us see next video we are going to see that reactions. So next one is just you observe the what is meaning by chlorophyll. You see here, we know very well in previous classes also we know something about chlorophyll. This is very important. And events occur in the chloroplast. So where does it take place? Photosynthesis very important, but where it does it take place means chloroplast only. Most plants, most algae and cyanobacteria perform photosynthesis. Such organisms are called photoautotrophs. Photosynthesis occurs in chloroplast in the plant cell, in the leaves of the plant. Inside the chloroplast is a green pigment called chlorophyll. So that captures light. Chlorophyll important function, it captures light. So let's see here color pigments in plants. So generally the principal pigments in plants are chlorophyll, carotenoids, xanthophyls, anthocyanins, betalains. So these are the pigments in the plants. Structure of the chloroplast. So here this is the structure of chloroplast: outer member, inner member, stomal lamella, stroma, lipid droplet, ribosomes, grana, starch granule. This is very important. And chlorophyll. So where does photosynthesis take place means chlorophyll only. So did not know how exactly location till Palatier and Cavallo discovered chlorophyll. It was believed to be spread in the cells of the green plants. So these you can study also. It is very easy. And coming to types of chlorophyll, very important. There are two main types of chlorophyll molecules. One is chlorophyll A, chlorophyll B. Another one is also there, chlorophyll C. Chlorophyll A found in all plants, algae, cyanobacteria. Chlorophyll B is an accessory pigment transfer the light it absorbed to chlorophyll A. So next one is chlorophyll A. Third type, chlorophyll C, is found in the dinoflagellates, brown algae or diatoms. Green light is reflected from the chlorophyll, giving the plants its green color. So next experiments, we are going to see some experiments. So some videos are also there, we are going to see in videos here. So starch presence in leaves, it is nothing but a presence of starch iodine test. This is very well known experiment also. So let us see. Tripod, hand, beaker, test tube, water, Bunsen burner. So methylated spirit, here very important procedure. Take a leaf. So a leaf of a plant which has soft and thin leaves and well exposed to sunlight. Boil the leaf in a methylated spirit over a water bath. Boil the leaf till it becomes pale white due to removal of chlorophyll. Spread the leaf in the dish and add a few drops of iodine. Observe the leaf. The presence of starch will be indicated by a black color. So here we have some precautions. Do not boil the methylated spirit test tube directly. Boil the water bath will low flame. So let us see these experiments. In this we are going to see, one is leaf experiment, another one is moles of leaf experiment and hydrilla experiment, and we are going to see here some experiments. So in video we are going to see this experiment. So here testing for presence of starch. So next, so here first we have to boil the leaf, because we have to remove the chlorophyll in that. So low flame. So after this we have to remove and take a test tube, the methylated spirit. So see here, we have to keep out the leaf, then take iodine drops, a few drops on the leaf. So it becomes, see here, dark color. So this is the leaf experiment. So presence of starch experiment. So next one is one more experiment we have, that is carbon dioxide is essential for the photosynthesis. So it is also called as moles of leaf experiment. So here carbon dioxide, moles of leaf experiments. Here the main important thing is potassium hydroxide. So potassium hydroxide, what is the use of potassium hydroxide? It absorbs carbon dioxide. So see here, insert split cork in the mouth of the bottle. So put potassium hydroxide or potassium hydroxide solution in the bottle. Insert one of the leaf of discharged plant through a split cork into the transparent bottle containing potassium hydroxide dioxide. So leave the plant in sunlight. So after a few hours, test this leaf and any other leaf of this plant for starch. So the leaf which was exposed to the atmosphere, air becomes bluish black, and the one inside the flask containing potassium hydroxide, which absorbs carbon dioxide in the bottle, becomes brown instead of blue black, showing the carbon dioxide is necessary for the photosynthesis. Here we can see. So again we have so far discussed the role of water and gases in the process of photosynthesis. So here what you get by this experiment, carbon dioxide is essential for the photosynthesis. Next, so one more experiment also there, that is Priestley experiment. So we know very well Joseph Priestley was discovered oxygen in 1774, performed. So he observed that when a burning candle is enclosed in a bell jar, the flame of the candle is gradually put off. See here. So he observed that when a burning candle is enclosed in a bell jar, the flame of the candle is gradually put off. In the same way, from this observation, Priestley conducted. See here one more. In the same way, a mouse also suffocates in a closed space, such as an see here. Such as in enclosed bell jar and dies. So next, from these observations, Priestley conducted that a burning candle and an animal change the composition of the air. See the experiment here. From these observations, Priestley conducted a burning candle and animal changed the composition of the air. He reasoned that something was necessary to the air. So Priestley then placed a mint plant inside the same bell jars, and he found that mouse stayed alive. This is very important. Mint plant. If you keep mint plants, this mouse was alive. So Priestley hypothesized that the plants produced a gas like oxygen and supported the burning of the candle and also allowed the mouse to survival. This is the respiration. Because of respiration, it survival. So air and photosynthesis. The production of the gas takes place due to the massive exchange of gas occurring through a particular section of the leaves called stomata and also through loose tissues and stems, roots. So this is the Priestley experiment. It is also a very important experiment. And next we are going to see light dependent reaction, mechanism of photosynthesis. Let us see mechanism of photosynthesis. So this is also very important. So photosynthesis basically light reaction and dark reaction. Light reaction, it is nothing but photolytic reaction. It is also called. Dark reaction means biosynthetic stage. So light needed to produce organic energy molecules like ATP, NADPH. It is also called assimilation powers. So dark reaction, no light needed. Instead, dark reaction use ATP and NADPH to produce energy molecules. This is the dark reaction. It is also called a biosynthetic stage. So light reaction. So light reaction, it is also called as light dependent reaction. It is mean light dependent reaction. So from the light and water. So here we are going to get from the light dependent reaction oxygen and as well as ATP, NADPH. By this ATP, NADPH, it is also useful to the light independent reaction, whereas carbon dioxide converts into the sugar molecules, or it reductase for carbon dioxide to carbohydrate molecules, means sugar molecules. This is light independent reactions. Next, light independent reactions. So let us see here. So this is chloroplast. So in the chloroplast we are going to see light dependent and independent reactions. The light reactions in the thylakoid membrane, it produces oxygen, ATP and NADPH. Here see thylakoid. So oxygen and ATP, NADPH. So ATP, NADPH, just now we have told about assimilation power materials. So next a Kelvin cycle, very important cycle. This is Kelvin cycle in the stomata uses of CO2, ATP and NADPH to make carbohydrates such as sugars. So here see CO2 and it converts into the sugar. So by the helping of ATP and NADPH, this is the dark reaction. So we know very well about TS of leaf, transfer section of leaf. So we have studied in previous classes also. Once see the video regarding this light dependent reaction, light independent reaction. Let us see here. So here plant cell, so chloroplast, so grana. So in grana we have seen stroma. In grana light dependent, stroma dark reaction. In grana light reaction. So in light reaction, so let's just observe chlorophyll in grana captures light energy. So after light energy, it splits water molecule into the H plus and OH minuses. This energy also useful excites the electrons, so it leaves chlorophyll. So after that energy used from the ATP, from the excited states, you just observe here diagrams. Then these H plus combines with electron, so here the reaction also there. So these hydrogen atoms reducing power for a dark reaction. So the dark reaction, so hydrogen ions and dark reactions. So next these OH minus ions, it means loss of electrons. This hydroxyl group, this OH combines into the H2O plus water, then it gives the release to the atmosphere in the form of oxygen. So we are going to see here. And next dark reaction. In this dark reaction, stroma, which is an important place of chloroplast. So in stroma we are going to see dark reaction. It is also called Calvin cycle. So just now we have seen the same thing in light reaction. After this, so this hydrogen atoms fix CO2. In dark reaction, CO2 is a major component. CO2 catalyzed by photosynthetic enzymes. This CO2 reduced from here glucose. So it is nothing but reduction reaction from carbon dioxide to carbohydrates or starch stored as such grains in the form of starch. So this is the light dependent and independent reaction. Next in this chapter we are going to see about the human digestive system. We know very well about in human digestive system. In previous classes also we have learned about the human digestive system. So here also we are going to a brief information about human digestive system. So let us see. So in human digestive system, mouth. So mouth ingestion, some mechanical and chemical digestion occurs. So we know very well about in this area, ptyalin plays a crucial role. That is salivary amylase in the form of ptyalin enzyme. So here it plays a crucial role. It also acts on carbohydrates. So that is a mouth. Next esophagus, from mouth to esophagus. Then stomach. So in stomach it is an important storage area. Mechanical digestion and chemical digestion occurs here. We know very well about the importance of HCl, hydrochloric acid. Here it plays a crucial role to digest the food material. And then next part is small intestine. So the majority of digestion occurs here only, small intestine. It plays a crucial role in digestion process. Chemical digestion and absorption occurs in small intestines, then moves to large intestine. So here also absorption of water occurs. And next rectum and anus. Digestion process also where we are going to see. This we already knows about this parts. So here there are some enzyme also plays a crucial role like ptyalin, pepsin, bile, amylase, trypsin, lipase, peptidase, sucrase. So one more one important bit also we can identify here. There is no enzymes in bile juice. This is very, very important. No enzymes in bile juice. So if you take pepsin, it acts on proteins, and amylase, it acts on carbohydrates. Trypsin, it acts on proteins. Lipase, it acts on fats. So sucrose we know very well about, in the form of primary, secondary, tertiary carbohydrates. It completely decomposes. Finally we have to reach the glucose molecules. So this is the human digestive system we are going to see here in 3D diagrams. So let us see here. Food. So from the food we know bolus. So bolus moves to the buccal cavity. So pharynx to esophagus, esophagus to cardiac stomach or pyloric stomach, then duodenum, then small intestine, stomach here, after stomach, see here, small intestine, then it moves to the large intestine. So here we have two important organs also there. One is the liver and another one is pancreas. So these can two attach here. So duodenum, the place where it is bile juice and pancreas. So after next one rectum. So here liver, gallbladder, pancreas. See the 3D diagram of a digestive system, how it is associated. Let us see. Just you keep on observing. So from food, mouth, buccal cavity, pharynx, esophagus, stomach, small intestine, large intestine, so that we have to need sufficient food material like vegetables and water. We can prevent our digestive system. So here see, so again it is going to come the same thing. So oral, pharynx, let us see, esophagus, stomach, liver is the biggest gland in our system, gallbladder very important, pancreas. So it moves to the small intestine, after that large intestine, then it moves to rectum and anus. This is the digestive system of the human. So next moves to the diseases. So we know some important some points also there, like vomiting, and one more thing, after vomiting we know what is the reason for vomiting. So next one is if anything like ulcers, stomach ulcers, this we can discuss later. Then we have to move. So malnutrition. So see here, there are three types of malnutrition occurs. One is caloric protein malnutrition, and calorie malnutrition, protein and calorie malnutrition. So very very important here. So expected bit from this. So Kwashiorkor disease, marasmus and obesity we know very well about. Kwashiorkor disease, it is a protein deficiency disease, and marasmus, it both protein and caloric deficiency. So marasmus, here we can see a diagram also. So obesity means it is over eating and over energy. So this is also one type of disorder. So we are going to see some vitamin deficiency disease we have studied in previous classes also. So we can study our own also. So let us see, there are many vitamins. So minerals or micronutrients, vitamins are all one area. These vitamins we have been classified into basically two types. One is water soluble vitamins, another one is fat soluble vitamins. So here water soluble vitamins is very important for the BC B. There are many vitamins in B group, B complex groups, B1, B2, B3, B4, B5, B6, B7, like B12 we have, and C vitamin very important, and fat soluble vitamins A, D, E, K. Once you go through this chart, we may get easy information. So what is the vitamins, what are the scientific name of vitamins, so what are the rich source for vitamins, so if we have the deficiency of vitamins, what diseases we are going to face, so what disease we are having, like that we have to see. This is about the nutrition chapter. So I tried my best level. So thank you one and all. If you don't understand anything, you just call me. So this is about the nutrition chapter. So thank you one and all. This video is dedicated to the all government teachers who are like us. Thank you one and all. 10th class students, let us discuss about the worksheets on today class. So we all know of about